Hi, my name is Carlos Brody. I'm going to tell you about a research effort funded by the National Institutes of Health within their BRAIN initiative, in which 7 different research groups have gotten together to work collaboratively to try to solve a problem that we think is too big for any single group to solve on its own. As a team, we call ourselves Brain Cogs, and you're going to hear 6 different snapshots of the work going on within this broader project. We'll start with an overall introduction from me, and then we'll dive into the 6 more detailed snapshots. We're interested in how working memory and decision-making are implemented in the brain. So let me take you through a quick example of working memory and decision-making. Suppose that you're playing a game of tic-tac-toe. I'm going to flash a screen of a game of tic-tac-toe of one turn, and I'm going to ask you, after the image is gone, to think to yourself, where would you play if you were playing O's? So here comes the image of the tic-tac-toe game. Okay, as you looked at that and then thought to yourself where you would play, you were using working memory. You were maintaining information as to what the state of play was, as to what the tic-tac-toe board looked like. You were manipulating that information as you thought, should I play my O here, or here, or here? And as you used that information held in working memory to consider available options, you committed to one as you decided where to play. How are these key processes, which we use so much in our everyday lives, implemented by neural circuits and neural dynamics within the brain? That's what we're interested in. There's reason to believe that many different brain regions across the entire brain are involved in these processes, and that's part of why we thought that we would need a team of different research groups working together to address the problem. Any one brain region can be very complicated, and interactions between many of them are more complicated still, but maybe together we can make headway. We use rodents in our research because there are many powerful neural circuit analysis tools that enable us to address the issue in great detail. So what are the groups that have gotten together? Let me take you through them. David Tank's group, Ilana Witten's group, and Sam Wong's group have all been involved in developing advanced recording and perturbation methods in rodents. David Tank's group is particularly interested in the function of the hippocampal formation and cortex. Ilana Witten's group is particularly interested in the function of the ventral tegmental area and the striatum, and Sam Wong's group is particularly interested in the role of the cerebellum in cognition. Sebastian Sung's group is leading efforts to link physiology and cellular resolution connectivity, recording the activity of many individual identified neurons, and then taking that very same tissue and figuring out exactly which neurons connect to which, so that with that information together, we can make headway in understanding how that neural circuit ticks. We also have 2 computational neuroscience groups, Jonathan Pilla's group, who lead efforts on advanced statistical methods to analyze neural data, and Mark Goldman's group, who leads advanced biophysical modeling of networks of neurons, both within and across brain regions. And finally, we also have my own group. We've been interested in working memory and decision-making in rodents for a long time. Now, I told you that we use rodents, sometimes we use mice, sometimes we use rats. Neither mice nor rats can play tic-tac-toe, so what is it that we actually do? I'll take you through 2 examples of the behaviors that we study in the rodents. In the first one that we call the accumulating towers task, we use mice within a virtual reality setup that was developed by David Tank's lab back in 2009. The mice are sitting on top of a styrofoam ball that is held up in a bed of air, and so it's very easy to move. And as the mice run and move their feet, the ball moves, and the motion of the ball is then used to drive a projection of an image on a screen that surrounds the mice. That way, we can have a virtual reality system for the mouse while the head of the mouse is held perfectly still. Holding the head of the mouse perfectly still is important because that means that we can use microscopy to look inside the brain and record neural activity while the mouse is behaving. So what's the behavior that we have the mouse do? As I told you, we call this the accumulating towers task. The mouse runs up the stem of a virtual T-maze, and as it runs up that stem, white towers appear to the left and to the right of the mouse. And when it gets to the very top of the stem, the mouse has to decide which of the 2 sides had the greater total number of towers. And it should turn to that side, the side that had the greater total number of towers. And if it turns to the correct side, it gets a little water drop as a reward, and then the next trial starts. So why did we choose that particular form of task? Well, because in order to do this, they need to use working memory and decision making. They need to accumulate how many towers they've seen at every moment in time as they go. That means that they're maintaining the tally of accumulated towers so far. And when a new tower comes up, they have to manipulate that tally by adding one to it, either for the left or for the right. So they have to maintain and manipulate information. And then when they get to the top of the stem and make a decision as to where to go, left or right, they have to use that information held in working memory to commit to one of the available options and make a decision. So this is a simple form of working memory and decision making that is nevertheless very quantifiable and that is quite suited to rodents. So that's what we use. Here's what it looks like from the mouse's point of view. As you can see, they run quite fast. They do this a lot. They do hundreds of trials per day. So they're very used to doing this, and they do it quite reliably. Now, we don't only use the accumulating towers task. We have other variants of the task that you'll hear about in the talks that come in a few minutes. Another variant of the task is one that we call accumulating puffs. And what I'm going to show you is that formally it's identical to this task, but it has different sensory modality as an input and a different motor response as an output. So that means that by looking at what's common across the two tasks, we can understand what are the fundamental cognitive aspects, and by looking at what's different, we can understand what are the things that are particular to the sensory modality that's the input, or the motor report that's the output. In the accumulating puffs task, the mice are stationary, they're not in virtual reality, and as they are stationary, their heads are also fixed, so we can use microscopy, and while they're there, they get air puffs, little pulses of puffs of air to the right and to the left whisker pads. At the end of the air puffs, they get a sound that is the go cue. They have to make their decision now. And their decision should be which of the two sides had the greater total number of puffs. And depending on that, they lick either to the right or to the left of a lick tube. And if they lick in the correct direction, they get a water drop as a reward. So as you can see, it's also accumulation of pulses of evidence. In the accumulating towers, it's visual evidence. In accumulating puffs, it's somatosensory evidence, the puffs on the whiskers. And the motor report here, it's not turning to one side or the other, but licking to one side or the other. So they share that formal structure, but they differ in sensory modality and motor report. All of the tasks that we use in this project are what we call the accumulating X variety. They all share that formal structure, and that's what binds all the different efforts within the project together. Now, as I said, we're taking an integrated brain-wide view, so there are many people in the project across the 7 different research groups doing experiments in many regions of the brain. There's also several people doing modeling and data analysis, and several people working on technologies that are used throughout the entire project. We're not going to tell you about all of the work that is going on. Each of these people leads an independent project looking into these processes. Today, we're going to focus on 6 different stories that are all part of this big project. And they're all tied together by all working on accumulation of pulses of evidence. Here's a picture of all the people currently working on the project. The 7 different group leaders that I introduced to you before are all based at Princeton University, except for one, Mark Goldman, who is based at the University of California at Davis. Our first speaker is going to be Dr. Lucas Pinto, who is currently a postdoc in the Tank and Brody labs at Princeton. One of the big problems that we face as we think of the relevant brain regions for working memory and decision making is identifying which are the relevant brain regions, both in terms of their neural activity and their causal contributions to the processes. Lucas decided to address this problem by using high-throughput methods to examine both neural activity and causal contributions across all of dorsal cortex for working memory and decision making. Lucas will be starting up his own lab at Northwestern University quite soon. For now, here he is telling you about the work that he did as a postdoc at Princeton.
Decision-making can come in many different flavors. For example, if you're driving in traffic on a clear day and you encounter a red traffic light, it is pretty easy to decide when to stop. But if it's raining torrentially out and if it's night, you might have a much harder time distinguishing the red traffic lights from the rear lights in the cars, such that you might need to stare a bit longer through your water splashed windshield before making a decision. So this very simple example serves to illustrate the fact that even when decisions have the same behavioral goal or the same appropriate response towards that goal, they can vary widely in terms of their complexity and perceptual difficulty. More generally, decisions can have many different underlying computations. So for example, on the clear day, low-level sensory processing of the red traffic light might be enough to trigger the appropriate action of stepping on the brakes, whereas on a rainy night, one might need to add intermediate time extended computations to accumulate the noisy sensory evidence that is coming in through your water splashed windshield. There are many other relevant computations for decision making. For example, another very commonly studied one is decisions that require working memory, that is, maintenance and manipulation of either sensory information or information about the action that the subject is required to take before actually executing that action. So in the experiments I'm about to describe to you, what we wondered was whether the activity patterns across many areas of the cerebral cortex changed when decisions required different underlying computations. More specifically, we hypothesized that more complex decision-making tasks involving working memory would result in more widespread recruitment of different cortical areas. So in order to do this, we trained mice to perform decision-making tasks while navigating in virtual reality. So more specifically, we trained them on 3 tasks that all occurred in the same T-shaped maze. And here their job is to run down the maze and turn either left or to the right to retrieve a reward. And across all these tasks, what informs where the reward is stimuli that the animals see as they traverse the maze, but across the different tasks, they're required to perform different computations on these very similar stimuli. So the more complex task that we trained them on was the accumulating towers task that requires the gradual accumulation of sensory evidence and therefore working memory. So as animals ran down this virtual T-shaped maze, they encountered these briefly appearing tall white objects, which we call towers, and their job is to count the towers because the reward will always be on the side corresponding to the highest tower count. We also trained them on a much simpler task that we called visually guided task, and this is virtually identical to the accumulating towers task. It also contains the same stimuli, but here additionally we place a visual guide, a tall visual guide that the animals can always see, and the guide indicates where the reward is. So this completely removes the need for evidence accumulation and working memory of the stimuli. We also trained them on an intermediate task that is called the memory guided task. And here the animals do not see towers, they only see the distal visual guide, but this guide disappears towards the end of the maze such that the animal needs to remember where it was. So the first question that we asked was what regions across the dorsal cortex were necessary for the animals to perform this task. And here we used optogenetic approaches to systematically, in an unbiased fashion, inactivate 29 different cortical areas, very briefly, only throughout the duration of the behavioral trials that were randomly chosen within the behavioral sessions. And so here we saw something very remarkable. What I'm showing you now is maps that summarize the effects of inactivating different cortical areas on the behavioral performance of these animals. And here what is important to note is that the red circles indicate areas whose inactivation caused significant decreases in performance. And so during the easier visually guided task that doesn't require working memory, what we noted is that primarily only the inactivation of these very few cortical areas that correspond to visual cortical areas that process visual stimuli affected the performance of the task. And in very stark contrast, when we looked at the 2 other tasks that require working memory, we noted very widespread effects of inactivating many different cortical areas on the behavior. Next, we wanted to understand what underlying activity patterns actually explain these large differences in the effects of inactivation. And to do this, we used wide field imaging to observe calcium activity over the entire dorsal surface of the cortex with spatial resolutions of tens of microns. And so the video that I'm about to show you summarizes our main findings here. Here, the activity of each pixel is color-coded such that the yellower it is, the higher the activity is. And this is a video from a mouse that is performing both the visually guided task and the accumulating towers task within the same behavioral session. And the maze here will just serve to orient you with respect to the timing. So specifically during the period while the animal is still experiencing the towers, what we saw in the visually guided task is brief early activation of those very same visual cortical areas that whose inactivation resulted in decreases in performance of this task. But again, in stark contrast, when this mouse was performing the accumulating towers task, throughout the accumulating period, that is, while they're gradually accumulating sensory evidence, we observed widespread activation over the surface of the cortex, and then we proceeded to show that this activity is indeed related to evidence accumulation computations. So taken together, these findings show that more complex tasks involving working memory recruit more widespread computations across the dorsal cortex.
You've just heard from Lucas Pinto about how our accumulating towers task appears to involve many different cortical brain regions. How is it that these different brain regions communicate? And what is it that they're saying to each other? Suppose you record from 300 neurons in each of 2 different brain regions. What are the shared signals between them? And what do those shared signals tell us about their communication? This is the problem that Dr. Stephen Keeley, who is currently a postdoc in Jonathan Pillow's group at Princeton, has been addressing. The methods that Stephen has been developing are general. They could be used in many different tasks and studies. We're, of course, particularly excited to apply them to our own task. Here's Stephen to tell you about his work.
Hi, today I'm going to tell you about some work I've been doing with Jonathan Pillow in developing statistical tools to find underlying structure in multi-region neural population data. I want to first draw your attention to the fact that over the past several decades, we've been able to record simultaneously from more and more neurons during behavioral tasks. Back in the 60s and 70s, we were only able to record from a handful of neurons, but now there are many neural data sets that contain 500 or even 1,000 neurons or more than 1,000 neurons. And this change in neural data underlines a need to develop new statistical tools to understand these neural data. And so today I'm going to talk to you about a particular statistical tool that I've been working on called a latent variable model. In a latent variable model, we describe high-dimensional neural data, so a large number of neurons who are described by a time series of spike trains for each neuron. We describe this high-dimensional neural data as having some low-dimensional structure. So we can see here this high-dimensional neural data set can be described by just an evolving time trace in just 2 dimensions. We can find these meaningful low description structures in neural data, and what this basically means is though we may have 100 numbers to describe the neural data here, we only have 2 numbers to describe, again, this entire population in this low dimensional representation. And these low D representations help us understand how neural activity represents information about the world. These are parsimonious descriptions, and they're also easily visualizable. So they're a useful way to make sense of this entire population of activity. In order to describe this, I'm going to step through statistically how we describe a single neuron first. So to describe a single neuron, we're interested in characterizing that neuron's firing rate. We do this by describing its average number of spikes per bin that we expect it to fire, and we describe that rate as smoothly evolving through time. So here I have an example of a firing rate of a neuron sort of smoothly evolving through time, and we expect this rate to give rise sort of randomly to action potentials that have a higher probability to fire when this firing rate is high and a low probability of firing when the firing rate is low. So we would expect an example neuron whose firing rate is described with this trace to have a set of spikes perhaps like this. Now again, this is a random process, but the number of spikes is proportional to the rate described above. Now, of course, we aren't interested in characterizing just the firing rate of a single neuron. These tools are developed to understand populations of neurons. So here I have 4 different firing rates describing 4 different neurons and their corresponding spike trains. We expect in our statistical methods to uncover low-dimensional structure for this population. So what do I mean by low-dimensional structure? Well, in this case, these 4 neurons can be decomposed into 2 functions, which I'm calling latent 1 or latent 2, or x1 and x2 here. By this decomposition, what I mean is that each firing rate is some particular combination of these two latent variables. So we can see here that this first rate is actually only a component of latent 1, but the second and third rates are weighted sums of latent 1 and latent 2. And because all of these rates for this entire population are different weighted combinations of latent 1 and latent 2, we describe this set of 4 firing rates as actually a two-dimensional population. So this is so these 4 spikes are actually two-dimensional population activity. Now, I'm now describing here exactly what I described in the last slide. Our central goal for these class of methods is to look at the spike trains and decompose it into, say we have these 6 neurons, decompose it into 2 latent variables that have some weighted combination. This is now a matrix operation describing a set of weighted combinations of these 2 latents describing the rates of each of these neurons. Because these are rates, however, we need to constrain them to be non-negative, so we put them through what's called a nonlinearity. This forces all of the numbers in here to be non-negative, and such that we have valuable firing rates, and we need that to be non-negative because firing rates cannot, in fact, for neural activity be negative. They have to be either 0 or positive. So again, the mapping is some latent variables via some linear combination of the, say, 2 latent variables put through some nonlinearity to keep the numbers positive, and that describes our firing rates. And this is a smooth low-dimensional latent space, and if you're curious, the mapping from the firing rates to the spike trains is via a inhomogeneous Poisson process. In order to extend this class of methods to multi-region data, I'll show you a schematic for how we do that. So here we have an example population from a certain region of the brain. I'm showing 4 example neurons from, say, region 1, and 4 spike trains from, say, region 2. In order to decompose this into its underlying latent structure, well, we actually decompose it into 3 different sets of underlying structures. So we assume some of the underlying latent structure that's low dimensional corresponds only to region 1. We assume some of the underlying structure that's low dimensional corresponds only to region 2. And we assume some of the underlying latent structure is actually shared between these regions. And again, this goes through a weighted sum or some combination, some matrix operation that's going to give me firing rates for each of these neurons, and each of these firing rates is going to map to so region 2 is only mapped, the region 2 latents are only mapped to region 2 activity. The shared latents are going to be mapped to both regions, and the region 1 latents are only going to be mapped to region 1. And again, all of these smooth firing rate curves are going to be described via Poisson noise to represent our entire neural population activity. And that is the general tool we are going to use to uncover latent structure that is both local to and shared between many regions in the brain. Thank you.
In what we've told you so far, we've mostly been thinking about cortical regions. But how about the rest of the brain? Dr. Marlies Oostland, a postdoc in Sam Wong's group at Princeton, has been thinking about this problem. And in particular, she has been thinking about the cerebellum. The cerebellum has long been thought of as a motor structure involved in controlling our motor acts. But in recent years, it's become more and more appreciated that it also participates in cognition, and Sam Wong's group has been one of the groups leading the efforts in this direction. In particular, there are parts of the cerebellum that are strongly connected with regions of cortex associated with cognition. Do they play a role in the accumulating towers task? Marlies has been studying this and is here to tell you about her work.
Hi, I will talk about the role of the cerebellum in decision-making, specifically how cerebellar representations evolve during learning of an evidence-accumulation decision-making task. Every day you need to integrate cues from your environment to make decisions, such as when you are crossing a street. You need to integrate the information about the cars coming from the right and from the left in order to make a decision when it will be safe to cross the street. You might think by now that this is easy to do so, but maybe the video of this dog will help you appreciate that's actually a learned behavior. You did not always know how to do this. So how does the brain actually learn how to integrate cues from the environment, and which brain regions could be involved? In Wang Lab, we're interested in the cerebellar processes contributing to non-motor functions, such as decision-making. We have developed an evidence accumulation decision-making task for mice in which movement is reduced. The mice are head fixed and sitting in a tube. The evidence that they need to integrate or accumulate over time are air puffs delivered to their whiskers. The decision they need to make is licking right or left. This is based on where the whisker puffs are given. If they receive more air puffs to their right whiskers, they will need to make a decision by licking right. After several weeks of training, the animals are doing very well at this task. So thus, if they receive more air puffs to the right side of the task, they will have a higher fraction of rightward decisions, which is correct. We've also shown that that cerebellar cruise 1 is important for the performance of this task. If we optogenetically and bilaterally inactivate cruise 1 of the cerebellum, the animals are back at chance level, which is indicated here by the blue line in a similar psychometric curve. So what goes on in the brain when the animal is performing this task? We have done somatic calcium imaging in Purkinje cells of cruise 1, and during the Q period indicated here by the gray box, some Purkinje cells ramp up their activity and other Purkinje cells ramp down their activity. The strength of this ramping up or ramping down, or the slope of this ramp, is correlated with how strong the evidence was during the Q period. We have now also replicated this finding with in vivo electrophysiology using silicon probe recordings. The higher temporal resolution this technique provides has shown us that there's also a sharp increase in firing rate at the onset of the Q period, which always starts with bilateral puffs. So the cerebellum is definitely not the only region contributing to Q period by increasing its firing rate or showing ramps up or down. This has also been shown for the VTA, the striatum, and also several areas of the neocortex are involved in decision-making tasks like this. So why does the cerebellum also need to contribute to this by showing ramps during the Q period? Well, the cerebellum has bidirectional connections to the frontal cortices. I'm highlighting the ascending pathway here in green. For instance, Purkinje cells in cruise 1 can project to the cerebellar nuclei, in this case the dentate nucleus, which projects to several nuclei in the thalamus, which can then proceed this information to frontal and parietal cortices. There are also many areas back. For now, I will highlight on the dentate nucleus and the role this plays in an evidence accumulation decision-making task. So when the animal is very well trained, there ramps up and down also in the dentate nucleus, similar to what we found in Purkinje cells in cruise 1. This is an example of one cell in the dentate nucleus of a trained mouse. Here are the parameters of the tasks explained. The task starts with an audio cue to indicate that the animal needs to pay attention. Then there's a cue period which starts and ends with bilateral puffs, and after a short delay, the lick ports arrive and the animals are able to lick and execute their decision. As an example from one cell through 1 hour training session and several trials. We see a sharp increase in relation to the audio cue, as well to the bilateral puffs at the onset and at the end of the cue period. Throughout this cue period here, we see an increase in firing rate, so it starts with approximately baseline firing rate and then increases. Also, we see a lot of activity when animals licking. We see this in several cells in the dentate nucleus, and indeed, some cells ramp up their activity, while other cells ramp down their activity. But what happens to these task-related dynamics when the animal is actually learning the task? What would happen the very first time the animal is in a setup? As I said before, when the animal is actually doing the train task, they have gone several weeks of training, and they have gone through 8 different levels of this task. In the very first session, the animals have never been in a setup before. They've never had an air puff delivered to their whiskers in this way, and it's also the first time they receive a water drop from the lick ports in this task. There's still an audio cue at the beginning. The cue period is much shorter, it's only 1 second now, but it can be followed by hints, which are air puffs to the whiskers on the correct side, which will continue until the animal actually licks. I will show you now this example of a dentate nuclear cell on the very first day of training. We see an audio cue, but there is not really visible response to the audio cue. However, there is a strong increase in firing rate throughout the first half of the cue period, and this increase is then followed by a slow decrease until it's back to baseline. Here, when the animal is licking, we actually see another increase. We also see examples of cells which do the opposite. So here, for instance, is an example of a cell in the dentate nucleus on the very first day of training that shows some indication of an increase in firing rate to the audio cue. During the first half of the cue period, instead of an increase, this cell actually responds with a decrease in firing rate, which then returns to baseline levels at the end of the Q period. Thus, we also see cells ramping up and down in the dentate nucleus already on the very first day of training. To conclude, I've shown you that in well-trained mice, both Purkinje cells in Crus1 and neurons in the dentate nucleus show ramps during evidence accumulation. And at the earlier stages of training, even on the very first session, there is already some indication of ramps in the dentate nucleus cells. Thank you.
In the behaviors that we've been talking about, our mice are motivated by water rewards. Are reward-related circuits also involved? This question led Dr. Ben Engelhardt, a postdoc in the Tank and Witten labs at Princeton, to wonder whether the ventral tegmental area, which is a region long thought to be involved in reward processing, is also involved in the accumulating towers task. To address this, he imaged from neurons in the VTA to look at their activity while mice perform the task, and as he will tell you, he found some very interesting results showing that not only was their activity correlated with reward, but also with many other different aspects of the task. Here is Ben to tell you about his work.
Hi, I will talk about dopamine in the brain and new experiments we are performing to understand the role of dopamine-releasing neurons in helping produce complex behavior. Dopamine is a molecule which acts as a key neurotransmitter in the brain. It is produced by a group of neurons called dopamine neurons, which are located primarily in the midbrain, a deep brain structure which has been conserved across species throughout evolution. These neurons then release dopamine to other important brain regions such as the striatum, as well as the prefrontal cortex. We note that dopamine is important for normal brain function because many brain disorders are characterized by dopamine dysfunction, most famously Parkinson's disease, but also others such as addiction, schizophrenia, ADHD, and many more. Correspondingly, we know from many clinical and pharmacological studies that there are many behavioral processes which depend on correct dopamine function. These include motor control, working memory, attention, decision-making, and many others. These findings raise a fundamental question, which is, how does the activity of dopamine neurons help mediate all these different behavioral roles? Previous studies that have looked at the activity of dopamine neurons during behavior have found responses mostly related to reward processing. Here is one representative study, which shows that many different dopamine neurons have very strong responses to reward delivery. Strikingly, these neurons seem to be responding in the exact same way. This has been a consistent finding across studies, which has suggested that dopamine neurons act as a homogeneous functional population, where all dopamine neurons do the same thing, mostly to respond to reward processing. Now, this picture does not explain how dopamine neurons could mediate the different behavior processes that we have discussed before. Therefore, we proceeded to perform new experiments which involve recording of dopamine neurons activity in a complex behavioral task. This task is a virtual T-maze task for mice, in which they run down the stem of the T-maze and they are presented with random cues either to the left or to the right. At the end of the T-maze, they have to turn to the side where there are more cues to obtain the reward. Here's how the task looks like from the mouse's perspective. Importantly, this is a complex behavior which involves many different behavioral variables, such as navigation in the T-maze, the kinematics or locomotion of the mouse, the reward cues, the accuracy in the task, the reward history, and the reward itself. Now recall that dopamine neurons are supposed to be a homogeneous functional population, where all neurons respond in the same way. But that is not what we found in this task. Here are all the neurons that we recorded in the task. I'm showing you the responses to the kinematics or locomotion of the mouse first. Color-coded is the response strength of the neuron, with brighter color indicating stronger responses. As you can see, some neurons have strong responses to kinematics, while some neurons have weak responses. The same pattern holds true for position. These are the responses for the position of the mouse along the maze. Interestingly, the same neurons do not seem to respond strongly both to kinematics and to position. We see the same pattern for the responses to other behavioral variables, such as the reward in the previous trial, the cues that are presented to the mouse, as well as the accuracy in the task. And so, in all these variables that occur during the navigation-based decision-making part of the task, we find a very diverse response profile, The different neurons seem to respond to the different things. We can also look at the responses to reward, which comes at the end of the trial. When we do that, we see a very different picture, where almost all neurons have very strong reward responses, consistent with the previous literature, which has shown that dopamine neurons have uniform responses for reward. We can also try to understand whether there is any structure to these responses. Do we have specific subpopulations which have similar response profiles? To do that, we group the neurons using a clustering algorithm. We find that 5 clusters explain best the data. When we order the matrix based on the clusters that we find, which are color-coded on the right, we see that each cluster is associated primarily with a single behavior variable that occurs during the navigation portion of the task. For example, neurons belonging to the red cluster are associated primarily with kinematic responses. Neurons belonging to the green cluster primarily with position responses, and so on. So we find that there are specialized subpopulations of dopamine neurons, that each of them responds to a different behavior feature or variable in the task, and all of these neurons have strong reward responses, consistent with the previous finding. We hypothesize that these specialized subpopulations could be a substrate that explains how the single molecule that is dopamine can help produce complex behavior and all the different behavioral processes that have been associated with dopamine in the past. And we are currently performing new experiments to test this hypothesis further.
Brain regions of particular interest for Ilana Witten's lab include not only the ventral tegmental area that you just heard about, but also the striatum. A postdoc in the Witten lab, Dr. Scott Balkan, and Iris Stone, a graduate student that is shared between the Witten lab and Jonathan Pillow's lab, have been working on data from the striatum. Their preliminary results suggest that animals go through bouts of very, very different states of behavior and of neural circuit dynamics as they perform the accumulating towers task. This suggests that we should analyze and think about our data very differently according to which of the different states the animal is in. Thus, although they have been developing their methods for data from the striatum, their results could perhaps apply across the entirety of the project. Here to tell you about Scott Balkan's and Iris Stone's work is Professor Ilana Witten.
Hi, I'm going to tell you about work from my lab and Jonathan Pillow's lab on the role of the striatum in evidence accumulation. So why are we interested in the striatum in evidence accumulation? So the striatum is, I think, a very interesting structure that's part of the basal ganglia, which is basically composed of it's part of these 2 parallel feedback loops onto cortex. One loop is called the indirect pathway. It's schematized with these yellow neurons. And the other is called the direct pathway. And what's interesting is that these 2 feedback loops provide positive and negative feedback to cortex. And this is interesting from the perspective of evidence accumulation, because these feedback loops may have opposing effects on the accumulation of evidence in cortex. So as a first step towards testing this idea, we wanted to inhibit the direct and indirect pathway during the towers task, first in the dorsal striatum. And what we found were huge effects from inhibiting the striatum, where you get opposite decision biases, with bias in the direction ipsilateral to the inhibition site if you inhibit the direct pathway, and contralateral if you inhibit the indirect pathway, which is consistent with the concept that these 2 pathways have opposing effects on cortex. And really interestingly, the same exact manipulation in the same animals, but on a T-maze with a visual guide rather than the evidence accumulation, did not lead to any bias in decisions, suggesting that this isn't a motor structure, but instead it's really involved in the decision-making process. So this says that the direct and indirect pathway have opposing effects on decision-making, but is this determined by the subregion of the striatum? If we did the exact same experiment, but in the ventral rather than the dorsal striatum, in that case, there weren't decision-making biases. There was actually very little effect. If anything, laser caused the animals to be a little less sensitive to evidence. So what this means is that the striatum has big effects on decision-making that depend on cell type and subregion. We thought this was really exciting, and to try to better understand these effects, we wanted to use computational modeling, both to better understand the behavior and better understand the effect of the striatal inhibition on the behavior. So specifically with Jonathan Pillow's lab, we've been using a generalized linear model combined with a hidden Markov model to discover discrete behavioral states during this task. For example, this is a schematic of the hidden Markov model, part of the model, where each circle here is a different trial, and you can think of different states where there's different probabilities of emitting a choice. For example, maybe in state 1 there could be a more probability of a right choice, in state 2 a higher probability of a left choice. And this is a hidden Markov model where the animal is more likely to stay in the same state than transition to a new state. And on top of the hidden Markov model, there's also a generalized linear model such that external covariates like the sensory evidence or the previous choice or whether the laser was on also influences the animal's choice. And the influence of these covariates depends on specific weighting that gets learned by the GLM, and which are different for different states. So this basically allows decision making to be modeled by a process where the animal is in different states at different periods in time with different weightings between the sensory and external covariates and the animal's choice. And what we find is that this model does a really good job explaining the data and discovers different behavioral states at different points in time. This is just one example session where the animal's briefly in state 1 early on in the session, but then spends most of the session in state 2. So what are states 1, 2, and 3? Here I'm showing you the psychometric curves from the same set of animals, but separated out by these states. And you can see that state 1 is a state with very low performance, where they don't do a lot of evidence accumulation, where they do the task much better in state 2 and state 3. And what's really interesting is these different states have different effects of inhibiting the striatum. And in fact, basically this suggests that the striatum contributes differentially to these different states and mostly contributes to this state 3, where the animals are doing a good job performing the task, but when you inhibit the striatum, their behavior gets really messed up, unlike these other states where the striatum is less involved in their performance. So what have we learned so far about the striatum? The dorsal striatum, if you inhibit, that generates a huge decision bias. The direct and indirect pathways have opposite effects on the animal's decision, whereas in the ventral striatum, you don't get a decision bias when you inhibit. So therefore, the effect of the striatum on decision making depends on pathway and also depends on subregion. Finally, the GLM-HMM suggests that the dorsal striatum contributes to behavior and generates these biases in specific behavioral states. Thank you very much for listening to us.
A typical neuron in the brain is connected to 1,000 to 10,000 other neurons. How do those connections shape the neurons' activities? Adrian Warner, a postdoc in Sebastian Soong and David Tank's lab, has been addressing this problem by working on a high-throughput system to first use imaging to record the activities of many neurons while an animal performs a task, and then use electron microscopy and semi-automated machine learning reconstruction methods to identify all the connections between the recorded neurons. As he will tell you about in the next talk, he has been developing this approach for a task other than the accumulating towers task. But the difficult part is the method itself, so we are very excited to see him develop it and to then apply it to the accumulating towers and accumulating puffs tasks.
For a mechanistic understanding of brain function on a circuit level, it is of fundamental importance to understand the direction of information flow in those circuits. Ramon y Cajal was one of the first neuroscientists that attempted to infer the direction of information flow from morphological reconstructions of neurons. He used light microscopy to observe the detailed morphology of Golgi-stained neurons and painted and reconstructed their shapes. In his tradition, we are building a functional connectomics pipeline here at the Princeton Neuroscience Institute that combines in vivo functional imaging with subsequent volume electron microscopy-based circuit reconstruction in the same specimen. The typical workflow is as follows. We start off with behavioral training and multiphoton calcium imaging in behaving animals. We then perfuse the animals and fix the tissue with aldehydes, stain the tissue blocks with heavy metals, and section the blocks into ultra-thin sections, typically 30 to 40 nanometer thin sections, that are then imaged in our high-throughput electron microscopy facility. The resulting petabyte scale datasets are then processed by our automated image processing infrastructure that also automatically reconstructs the dense morphology and synaptic connectivity between those neurons using deep convolutional networks. Finally, we map the measured multiphoton calcium activity onto the reconstructed neurons in order to perform a detailed function-to-structure analysis. In one of the ongoing functional connectomics projects here at PNI, we aim to reconstruct the neural circuit underlying sequential neuronal activity in retrospinal cortex of mice performing different working memory tasks in a virtual reality setting. On the left, you see an example of a delayed match to sample with two alternative forced choice tasks. When the trial starts, the mouse runs along the linear track and enters a sample period in which a particular pattern, in this case a vertical stripe pattern, is shown on the walls. As the mouse continues running, it enters a delay period in which the sample is switched off. When the mouse continues running towards the end of the linear track, two arms appear, and the mouse has to turn to the one arm that has the same pattern on the walls as has been previously shown during the sample period in order to get a reward. We image the calcium activity of hundreds of neurons simultaneously in different cortical layers while the mouse is performing this task. Thereby we find sample-specific sequences of neurons that are active throughout the whole experiment. The sequences during the delay period, however, are of particular interest because we think they represent and correspond to some form of working memory. Different circuit models have been proposed that could generate this kind of neuron sequences. The most famous one is probably the SynFire chain, in which layers of neurons are connected in a feed-forward fashion in order to propagate neuronal activity from layer to layer. Alternatively, the neurons could be arranged in a so-called ring attractor network, in which combinations of local excitatory connections between neighboring neurons, long-range inhibition and asymmetric connectivity results in bumps of activity that move in a temporal ordered fashion. Similarly, random networks can be trained to generate these kind of sequences by modifying only a small fraction of the overall synapses. In order to validate or falsify any of these models, we need to measure the activity of hundreds of neurons simultaneously, and we need to reconstruct the dense connectivity between the neurons. For this, we apply a functional connectomics approach. Volume electron microscopy is the only technique that has sufficient resolution to resolve each individual synapse and can cover sufficiently large fields of view that contain entire local cortical circuits. We therefore are building a high-throughput transmission electron microscopy facility here in Princeton, consisting of several transmission electron microscopes with a collective image acquisition rate of several terabytes per hour. This is sufficient to acquire high-resolution stacks of cubic millimeter-sized tissue blocks on a regular basis. On the right, you see such a large section of a piece of mouse retina that has been imaged in less than 10 minutes with one of our transmission electron microscopes. As you can see, the resolution is sufficient to even resolve ultrastructural features such as endoplasmatic reticula, mitochondria, their cristae, and of course synapses and their vesicles. Processing and reconstructing these petabyte-sized image stacks requires specialized image analysis tools. Our lab has developed deep convolutional network-based automated segmentation algorithms that automatically reconstruct the morphology of the neurons and the synapses in these large electron microscopy stacks. Here you see a subset of 360 pyramidal cells that have been reconstructed from a large electron microscopy dataset that has been acquired by our collaborators at the Allen Institute for Brain Science. These neurons are from layer 2-3 in mouse visual cortex. With these automated analysis tools at hand, we can now start performing detailed functional to structure analysis of entire local cortical circuits. For example, we can look at the higher order organization of excitatory to excitatory connections in layer 2 and 3.
We hope you've enjoyed listening to these 6 different snapshots of work going on within the broader overall project as we strive to understand working memory and decision-making and how they're implemented in the brain. Each of the snapshots has its own motivation, its own person or persons leading it, and its own development, yet they're all closely bound together by using tasks that involve accumulating pulses of sensory evidence, and therefore they all inform us about working memory and decision-making. We didn't tell you today about our current efforts to develop models, both statistical and biophysical, that integrate information across many different experiments and many different brain regions. Developing such models is one of our important long-term goals, and each of the snapshots has contributed fundamentally towards that goal. Thank you again for listening.